This video was made as a guide. It does not replace training in any way. This is if you forget something or you need a quick review. Never do this or attempt to do this unless you've been properly trained by the proper person.
Okay, well, I'm going to tell you a little bit about the optical microscope. We won't be able to do a full teaching session, but at least identify some of the parts and some of the precautions. This is, of course, the optical microscope. There's a lamp back here with a simple knob that you increase the intensity of the light with. It's a white light source, an incandescent source. The inner lab has a gas source which has a lot more of the spectrum in the blue, which is good for very high resolution imaging. But for most routine optical microscopy, this source is fine. This is the stage, of course, and the first thing you want to do is make sure that the stage is in a down position before you start rotating any of the lenses. Now, if the person, this is a whole turret with multiple lenses on it. The smallest, let me make sure this stage is way down here, because the one thing you don't want to, one absolute precaution is you have to make sure you don't smash the lens into the stage. This is the turret of lenses, and they're, as I mentioned, they're 10x, 20x. By the way, I cautiously moved the stage down before I rotate it. So 10x, 20x, 50x, 100x. Oh, and by the way, the total magnification of your image is the product of the magnification here, which in this case is 100x, and the magnification of the eyepiece, which is 10x. So I'm seeing effectively 1000x magnification when I have the 100x lens here, because I have a 10x eyepiece. So you always want to start with the turret in the lowest position with the lowest magnification lens down. Never leave the system with the highest mag lens in, because those are the most expensive lenses and the most vulnerable. So we start with the 10x lens, and I move the XY stage so that the thing I want to look at is pretty much lined up as best I can, and then I bring the stage up. There's a knobs here that raise the stage to within approximately the focal length of the 10x lens, which you don't know yet, but you can sort of tell from the way the light is hitting the sample that you're looking at. So I'm going to now take off these glasses and look at what's happening here. Okay, so I have it not too far from being perfect focus, and in order to determine when I'm really in focus, I use one of these knobs here. The very first knob that I come to is called the field diaphragm. Basically, it's just a little, what happens is when you're looking through the microscope, you see this aperture that you can open and close. It's called the field diaphragm, and that's imaged on the sample surface. So when you look through the lens, you can see this aperture diaphragm. So I move the stage up until the aperture diaphragm is in good focus, and when it's in good focus, the sample is in good focus. Okay, so now I have the lens, the 10x lens focused on the sample, and this turret is set up to be what's called parfocal. That means that as I rotate from the 10x to the 20x is going to be out of focus, but it's going to be in such a way that I have to raise the stage to become in focus. So sure enough, I have to raise the stage just a little bit to get in focus. And now I'm going to switch into the 50x lens carefully, and the 50x lens should be out of focus or very close to focus, so clearly you can see which way. Clearly you can see which way to go. Ideally, it should be in a situation where I have to raise the stage to put it in focus. Now I'm going to switch to the highest magnification and most expensive lens, the 100x. And this is definitely out of focus, and I have to adjust it a little bit, and there I have it. So I'm using now by going through that sequence of steps from 10 to 20 to 50 to 100x, I'm able to look at the highest magnification. Now, you notice there's a little attachment on these lenses. What's inside there is a prism. It's called a Wollaston prism, because these lenses are suitable for doing what's called differential interference contrast, or what's often called Nemarsky image contrast. It's not strictly speaking Nemarsky, because that uses a different kind of prism. This is a special kind of differential interference contrast that's invented or installed by the Leica company. Okay, so in order to utilize the differential interference contrast, you have to have two things. You have to have a polarizer. This is an insert to the microscope. This is a polarization with the axis of polarization as indicated here. And the next one, the axis of polarization is that, and this is no polarization at all. So if I put this in, the first detente, so I have one of the polarization filters in, and the polarization filter is essential in order to utilize the differential interference contrast. And then there's this knob here, which I can twiddle, which is basically it's an analyzer. It's another polarizer, but it's set up in a different portion of the system. What I mean is this rotating polarizer here is on the illumination, because the illumination comes in like this, goes down to the sample, comes back, and passes through this filter. So these polarization filters do complementary functions. One is in the illumination path, this path, and one is in the imaging path, which is this path. What it really enables you to do is to visualize with the eye the topography better with this by twiddling this polarizer. I could explain how it works, but basically the system, the differential interference contrast, enables you to have two images, one for this polarization and one for another polarization, and when they pass through the analyzer, these two orthogonal polarizations can interfere. And that's basically what's happening. Okay, I can go to the other polarization here. That makes a little bit of difference. It's not possible to go through the whole thing, or even for operationally, you don't have to really fully understand how it works. The important thing is that you can get various contrasts as a function of the position of these filters and that's called the analyzer, the polarization knob, the rotation. Okay, there's another knob which most people are confused by, but let me point out where it is. This first little knob here, this first knob, that's the field diaphragm, which I've already explained. But the second knob is a little bit mysterious, but I'll tell you what it is. It's called the aperture diaphragm. And notice that this knob is in the illumination path. And what it does is it adjusts the angle over which the angle with which the light comes into the object being studied. So if you want to illuminate it with a plane wave, with a vertical illumination, you actually close this down, okay, and it also reduces the amount of light. And sometimes you can get some interesting edge effects when you do that, because there's sort of like diffraction effects from the edge. But you see things appear a little bit different. But if you want to get the highest resolution, you actually have to take this aperture diaphragm and open it up maximum, because that maximizes the angle of incidence of the light on the sample. And for reasons that are explained in the course, that gives you another factor of 2 in resolution, and enhances the resolution. I'm going to switch in the dark field lens. The dark field lens should be confocal, and it's pretty much in focus itself now. It's also confocal, and I have to adjust the focus a little bit, and here I have it. In order to use the dark field lens, you've got to pull out all these inserts here and put the dark field lens in position, and you've got it. And unfortunately, would have to show you basically what the dark field lens does is it accentuates things like dust particles and edges, sharp features, because it scatters light. And the flat surfaces look black, and anything that sticks up from the flat surface looks bright. And so it's a very good way for inspecting a surface for dust particles, for example, or roughness.